want to thank organizers for inviting me to present on this IGB Symposium, which happens finally, because I was actually supposed to present this work last year, but now it's postponed. So in Maslov Lab, we actually work a lot on consumer resource modeling of various microbial communities. And today I'm going to tell you about our recent efforts on bringing our intuition to a human gut microbiome case. As many of you probably know, a human gut microbiome is one of the best studied ecosystems because obviously it's related to human health. So people were collecting lots of data in the past decade, and they figured that microbial community in the gut is actually very complex, it's very diverse, it's dense. And what it typically does, it consumes dietary fibers which are undigestible by human body and converts them into myriads of metabolites that participate in various cross-feeding interactions between those microbes, and which are also can be consumed by human host cells. And through this mechanism, they actually affect our health a lot. And if there is any disruption in production of certain of such metabolites like short chain fatty acids or secondary bile acids or some vitamins and amino acids, it may result in very severe health problems and various metabolic diseases. Recently there was a shift in this field where people realized that if you want to characterize this gut community, it's not really enough to collect just a data set on microbial profiles. We also would like to know what metabolites are out there. And while microbial profiles were collected for quite some time already using metagenomics or 16S sequencing, and people developed really standardized protocols, and the cost of sequencing went down. So we actually can measure microbial profiles pretty reliably. It's not the case for metabolomics, because for metabolomics measurements, it's typically very hard to measure the whole set of metabolites which are out there. It's expensive. We don't really have standardized protocols. And if you go out there and look at different studies, they typically measure different sets of metabolites. So it's really hard to compare those things between studies. And the whole venture here is how to combine those two types of data to get some sort of mechanistic understanding of what's going on in this community. And we see it as another kind of challenge. Is it possible to actually predict metabolites from microbial profiles using discovered knowledge about those metabolic interactions? So when we first started to think about this problem, we stumbled upon this database that were published recently, and it keeps being updated, where people collected this knowledge about most prevalent human gut microbial species. We worked with like 600 of them, and they specified what those metabolites can actually consume and what they can produce according to various experimental studies. And this is a nice first step to start building a mechanistic model we desire. But the problem is that if you want to build this model quantitatively, we also have to specify a lot of numbers on top of this consumption production network. So consider the following situation. Imagine that you have two microbes that compete for the same metabolite, and in this network it's specified that they both can consume a certain chemical. How to split this chemical between those two microbes is not a trivial thing. And most intuitive and naive assumption would be is that if you have different abundances of those microbes, each microbe will probably consume this metabolite in proportion to its abundance, because you can expect that if you have a big population of a certain microbe, it will uptake this metabolite much faster than another one. And eventually it will just grab its proportion of metabolite according to its abundance. And this is what we actually do for our model. And then comes the second step, when a certain fraction of this metabolic intake comes into biomass and a certain fraction goes to byproducts. And for byproducts, if you have several of them, you have the same problem, how to split this mass between different byproducts. And the most simplified way, again, we just split it evenly in proportion. In this example, when we have two different byproducts, we just split them 1-to-1 in proportion to abundance of bacteria that generated them. And one step of a flow like that, when we have consumption of certain metabolite, growth of bacteria, and production of byproducts, this is what we call a trophic level. And the whole model for a community works like this. We first grab a set of metabolite in the database that can't be produced by microbes, but are in turn consumed by a lot of microbes. Those metabolites are typically out there as a dietary intake. So we grab those dietary intake metabolites, we assign them a certain amounts, and then allow microbes that can consume them to grow on them and produce byproducts. Then we go to a second step. We grab byproducts and allow microbes that can grow on those byproducts, consume them, produce a second round of byproducts, and so on. And on each of those steps, we actually will have a set of metabolites that are unconsumable by microbes. And this is what will be left as a metabolic output of the community eventually. And this is what we call our metabolome in the model. And we can actually go and compare those sets of metabolites to real measurements. And a couple of challenges that exist if we try to do a model of human microbiome like this are the following. First, we don't really have measurements of gut nutrient intake. Typically, when people collect all those omics studies, they just measure microbial abundances. If we're lucky, they also measure metabolite abundances, but that's it. But this nutrient intake is crucial for a model. So we actually employ machine learning procedure where we try to fit this nutrient intake in a way that it will best explain observed microbial abundances across all patients in our cohort. Another thing that we have in this model is that we will have to specify those parameters that I mentioned already. One is a fraction of consumption that goes into byproducts on each level. And another is the number of those levels that we want to propagate our models. And we actually also use real data for many patients to feed those two parameters. And we found a sweet spot, which is 4 trophic levels and byproduct fraction of 0.9. That gives us the best prediction when we compare our real measurements for the patients and values fitted with our model. So as you can see here on the right, this is an example for one patient in our cohort. We have a pretty good agreement for microbial measurements. So those red points are really close to the diagonal. Then we have a slightly worse prediction for metabolome, but still, assuming that we have so many simplifying things in our model, it's still great that we were able to predict metabolites on the level of like 0.67 correlation with this simplistic approach. And we also observed like several general trends when we apply this model to a big data set of healthy individuals. One is that consumed metabolites goes down as we propagate this model, which is expected, while unconsumable metabolites are accumulated. Another kind of expected trend is that higher trophic levels are much more populated and microbes there have higher abundances. And this is what we reported back then in our PLOS Bio paper. But actually, after that, we haven't stopped on that because obviously there are lots of limitations in this simplified trophic models. One is that, first of all, we were pretty discouraged that we can't predict many metabolites that are measured out there in all those untargeted metabolomic data sets. Right now, model can predict only a set of metabolites that were defined in the underlying network that we grabbed from the database. Another limitation is that because this database based on the previously published research, and we haven't really studied all microbial species in human gut in details, we definitely missing a lot of production and consumption links in this network. So we have to fill them in somehow. And then another thing is that this accuracy of 1.67, although it's comparable to other machine learning techniques that are out there on the market, it's still not really high enough to make individualized predictions for different patients. So we thought, like, is there any way for us to do better? And this is how we arrived at our second phase of this project, where we try to improve the model by using all those omics data sets to actually improve our underlying network of the model. And the idea goes as follows. Assume that you start with original network and you have a certain level of predictions across all individuals in a cohort. And now you want to improve it. You start adding links to this network under assumptions that probably those links are missing. And you start checking if a certain link that you added improves your predictions across all patients. If it is, this link is probably out there, but was not mentioned in the original database. So we should add and keep it in our model. However, the obvious pitfall here is that what if you have some false positive links that like somehow improves your prediction for a given set of patients just by random for some obscure reasons. And we thought about it and employed statistical approach to overcome it. It works as follows. We run this link addition algorithm for multiple individual runs. And on each run, we throw in random links in random order and keep those that improve our prediction. In the end, all runs converge to a certain number, which is our best reachable prediction. And then we look at the statistic across all of those runs and see which links pop up much more often across all those ranks. With the idea that if a certain link was added in multiple individual runs and improve prediction in all of them, this link is probably something really important and we should add it. And we filled 300 metabolic links using this procedure. And then we went and actually checked if there is any genomic evidence that those metabolic capabilities are out there for those microbes. And it turned out that 65% of those links that we predicted by our machine learning dump procedure are actually supported by genes present in those microbes. So this was very encouraging, and we keep using this procedure and expanding our network. And if you look at the individual predictions that we were able to obtain using this procedure, they actually become much better in the following sense. First of all, before, when we had our microbial predictions, for certain microbes, we had no consumption links for set of metabolites that are present in a given patient. And then those microbes are not really there according to our model, while they were measured in 16S or whole genome data. Well, now we added those consumption links to those microbes. They were able to survive. So all those points on the left here, they were brought to the diagonal. Our metabolite predictions also became better, and we actually reached 10% improvement in our accuracy. It went from like 0.67 to 0.75. And even more important part of this proposed machine learning procedure is that we were actually able to expand the list of metabolites that our model can predict, because now we can add production links to those metabolites and use them to predict their levels. And here on the right, you can see that we actually expanded list of metabolites here like 2 times, and we can predict them reasonably well. And so to conclude, we actually now have this trophic model that approximates metabolic flow through the human gut microbiome. And using this model, we can predict levels of some metabolites using microbial abundance data, which was measured by 16S and whole genome sequencing. We also developed a method to improve this model by proposing new metabolic links and comparing them to existing data sets. And we actually continue actively working on this project because there is some room for improvement. 0.75 is not like 90% accuracy, right? So we plan to collect more multi-omics data sets. So if you have a data set like that, please contact us. We will be happy to collaborate. We also are trying to fine-tune consumption and production parameters, because remember, right now we have this global parameter for byproduct fraction for each of the microbes, and we are also splitting those microbial consumptions in proportion to abundance. We also split in byproducts evenly, but we all know that there is some stoichiometry, and we probably have to take it somehow into account, and it might actually improve our predictions even further. So we'll keep working on it. And this is our project team. The main authors of 2 papers I discussed, and also we now have some additional students that join this group and keep working on it. And with that, I want to thank you for your attention, and I'm ready for any of your questions.